3-week push to the end of the semester. I have a couple of announcements before we get started. One is that this Thursday there's a senior seminar presentation in the biology department on ecological factors that influence sociality in bees, canids, aphids, and shrimps. Who knew that shrimps were social? So that's this Thursday, and then next Thursday is another senior seminar related to electrical activity in the heart, something that you're familiar with, jellyfish, and plants. Who knew that plants have electrical activity? So those are senior seminars coming up this Thursday and next Thursday. So you've received your exam 3 back. The key is up on Moodle. Feel free to ask me after you've looked at the key, see where feel free to ask me questions about what I was looking for if it isn't clear. I didn't spend a lot of time writing on your papers, comments, because it was taking a long time to grade as it was. But I'm happy to give you my feedback about what was missing. Sometimes there's some cryptic little notes in your answer that might give you a clue. So today we're going to continue our journey down the alimentary canal. Focus on the stomach. So remember there is an anatomy quiz. Well, it's a quiz that's due this coming weekend. That's Sunday night at 11:30. It will be available probably later today at the CST desk. It will cover material through Thursday that we cover in class and is assigned in the coloring book. So the coloring book and class material is fair game for this quiz that is due on Sunday. So today our goal is to talk about the stomach from an anatomical perspective and also from a cellular enzymatic perspective. That is, names of things is not just gross anatomy, but includes names of hormones and what they do. So we'll talk about the gastric acid in the stomach, pepsin, and if there's time, we'll get to ulcers of the stomach, gastric ulcers. So let's start with a clicker. Let's activate the room here for a little bit. Let's do a clicker related to. Well, this is related to getting food from your mouth into your stomach. Okay, I'm seeing a lot of partially correct answers, but I don't think I see a single fully correct answer because the first 3 are correct. You certainly, chewing mixes it with saliva, so A is true. Carbohydrate is enzymatically hydrolyzed by the amylase in saliva, and lipid is enzymatically hydrolyzed by the lipase. Lingual lipase is one of the enzymes in saliva. Protein is not yet enzymatically. It might be mechanically broken up. I mean, you chew a piece of meat and the protein is getting chewed up, but it's not. There's no enzyme in saliva that is a protease. Okay, so let's do the next one. You swallow. What happens? Okay, in this case, I'm seeing several fully correct answers. A and C are correct. There's a reflex that for swallowing. It's a reflex. You're born knowing how to do it for swallowing. Not for chewing, but for swallowing. Chewing is something that we learn how to do. Swallowing is something that we're born knowing how to do. The epiglottis closes. That's the closing of the epiglottis is what prevents the food from being aspirated into the wind, into the airway. Food is propelled into the stomach by esophageal peristalsis. Yeah, we saw that on the video, and we'll see that again in a moment. We don't need gravity to get food into the stomach. Astronauts swallow their food just fine when they're up in space. So I want to review two videos. One is this one, the one that we saw before break.
Swallowing, or deglutition, is the process by which food passes from the mouth to the stomach.
That action right there is peristalsis. A wave of squeezing down the esophagus that propels a bolus of food down the esophagus into the stomach. That is what peristalsis is.
This movement is controlled by the nervous system and involves both voluntary and involuntary muscle contractions. Before food is swallowed, it is mechanically broken down in the mouth by chewing or mastication. The salivary glands secrete the enzymes lingual lipase and salivary amylase.
Lipid and carbohydrate enzymatic processing.
Salivary amylase begins the chemical breakdown of starch, carbohydrates, in the mouth. The water content of saliva helps bind particles together to form a bolus that can be swallowed. Swallowing occurs in 3 stages. The first is a voluntary phase called the buccal or oral transit phase. Here the tongue moves upwards and backwards against the hard and soft palate to push the bolus into the oropharynx. Next is the pharyngeal stage, in which involuntary movements push the bolus through the pharynx and into the esophagus. Movement of the bolus stimulates receptors in the oropharynx, which send impulses to the deglutition center in the medulla oblongata and the lower pons of the brainstem. The brain signals the soft palate and uvula to close off the nasopharynx, and the epiglottis to seal off the larynx. This prevents the bolus from entering the respiratory tract.
Closure of the epiglottis.
The upper esophageal sphincter relaxes to allow the bolus to move into the esophagus and contract to reduce backflow into the pharynx.
So here we're seeing the peristaltic squeezing of the bolus of food down the esophagus.
In the final esophageal stage, the bolus is pushed onward by peristalsis, a progression of involuntary wave-like contractions of the circular and longitudinal smooth muscles of the esophagus. As the bolus approaches the end of the esophagus, the lower esophageal sphincter relaxes and the bolus moves into the stomach. So, in summary.
We've summarized. Let's look at actual data. So this is an X-ray of someone chewing something that has barium or something in it to make it X-ray opaque so that you can see it being moved in the mouth by the tongue and the jaw, and then being pushed by the peristalsis in the esophagus. See it being squeezed. Now he or she is going to work on a liquid. a little fast, but let's see if I can slow it down. That's not So let's just do one of these side views in slightly slower motion. I don't want to go down this side. So you can see it's not just falling, it's being pushed by the peristalsis down the esophagus. It's being squeezed down the esophagus. So you can look at these some more if you're interested. So now, let's talk about the anatomy of the stomach. So here are some names that you should learn for the quiz. The esophagus, the lower esophageal sphincter connects the esophagus to the stomach. The stomach has regions that are given names. The fundus in the superior region of the stomach. The cardia is the area right where the lower esophageal sphincter is, where the esophagus joins into the stomach. So this ring right here is called the cardia. The fundus, the body is the largest area of the stomach. The antrum is this area here. The pylorus and the pyloric sphincter are at the far end of the stomach. The region of the small intestine that the stomach connects to is called the duodenum, the duodenum. Your book makes a point of saying what the body secretes and what the antrum secretes, but since the border between the body and the antrum isn't very distinct, what they secrete, I mean, the differentials of what they secrete is not that important. They both secrete, we'll talk about their secretions. Mucus, precursor enzyme to pepsin, called pepsinogen. So if you see ogen on the end of a word, it's a precursor. Just like angiotensinogen was the precursor to angiotensin. Now we're going to see that a lot. Pepsinogen is the precursor to pepsin. So what is secreted in the stomach is pepsinogen, which is then converted into pepsin, which is an enzyme. And acid. Stomach acid, gastric acid, hydrochloric acid. It's all that's gastric acid, hydrochloric acid. And the antrum secretes basically the same stuff. Okay, so there are some of the names. Here's another, okay, you remember this game. So let's play this game. What's that? Oh, handout. Is there some more handout?
Anyone else?
Let's play this game here. Who wants this? Let's just start. Okay, so pick a dot and give me a name, and we'll let the Bing tell us whether it's right. Okay, that's the duodenum. Is the laser failing? It might be failing. The batteries might be running out. In which case, I can give you this one. The fundus? Here, right here. So the middle button is the laser button. Okay, so you're, I think, so you're saying the fundus is one of these two. Let's go with this one. That's the fundus. Why isn't it updated?
This is not the website.
It's very strange. I mean, what I'm seeing, I see it move and I see it,
It's a slide, yeah.
oh, because we're still in the PowerPoint, but we're not on the. Say again? Esophagus. This one here? Ruby. You want Ruby to be here. I don't think so. No. So just this, what we're looking at here are muscle layers on the outside surface of the stomach. Yeah, those are illustrating the gastric ruby, the folds on the inside of the stomach. So the cartoon is maybe not so clear even here. I mean, this is looking at the inside surface of the stomach, and this is looking at muscle layers on the outside surface of the stomach.
So in the hangout, they spell Ruby R-U-G-E-A. This one spell A-E.
R-U-G, oh, I spelled it wrong. This is right. R-U-G-A-E. This is the lesser curvature, yes. So that's something that wasn't on the previous slide, but it is an anatomical name. This curvature of the stomach is the lesser curvature. This is the greater omentum. So this layer of fat, so the lesser omentum connects to the lesser curvature, and the greater omentum, which is a flap of fat that connects to the greater curvature of the stomach. So the names are somewhat helpful. Okay, so now we're getting down to some other names.
Is this the bottom?
Yeah, so, well, I think this is this the one you were pointing to?
Yeah.
Yeah, that's the body. We've got a 50-50 choice here. Yeah, this is the circular muscle. So we have layers of muscle, longitudinal, circular, and oblique layers of muscle. This is the circular muscle. And what this is pointing to is the outside, it could have pointed anywhere here, is the serosa, is the name of the outer layer of the stomach. Okay, so there's another one here. Let's do some more. So keep passing. Who's got the laser? Mallory, you want to choose one and give me a name? Yeah. Pyloric sphincter is this muscle at the bottom, far end of the stomach. Can you help me if I dim the lights a little? Does that help at all? Which one do you? Body is not one of the options in this one. Okay, which one is the greater curvature? Right, so that is the greater curvature. Greater omentum is connected to the greater curvature of the stomach. The middle button, there, it's where,
This is the
right. So the diaphragm, you know that from the previous section of the course. The diaphragm is that thin sheet of muscle that separates the lungs from the abdominal cavity. It's the muscle of respiration.
This the thyroid canal.
Yeah, so, I mean, you know, pyloric sphincter is a canal. Pyloric? Well, that's weird. Okay, so this is the pyloric canal. That's weird. This thing is calling this the pylorus. Okay, don't worry about it. This is the pyloric sphincter. Okay, who's got the, yeah. Yeah, so the antrum is the region, so we have the antrum, the pylorus, and the pyloric sphincter. That's right. So this is the oblique muscle, and now I'm going to deprive one of you of an easy answer. This is the longitudinal muscle. Okay, so now,
that's the lesson.
right, so this is the lesser omentum connected to the lesser curvature, and this area right here is the cardia. Okay, so these links which are available to you on the PowerPoint, you can use to test your knowledge, or you can use the coloring book, or any other resources for learning these names for the anatomy of the stomach. So this is the PowerPoint. Okay. Yeah. So anyway, the point is, at the top of the slide are the links to those things, and you can just cut and paste them from the PowerPoint, which I'll post to Moodle if you want to play the game at home. Okay, so we saw motility in the esophagus. There is motility in the stomach too. So the bolus of food gets squeezed from the esophagus through the lower esophageal sphincter into the stomach, where it is now digested by mechanical and by chemical action. The chemical action includes the acid of the stomach, the gastric acid, the pepsin that we're going to talk about in just a minute, that enzymatically hydrolyzes protein, and motility, that is to say, a churning action, squeezing action of the stomach. The stomach is a muscle, and so the circular, longitudinal, and oblique muscles contract to make waves of peristalsis that squeeze the chyme in the stomach, churning it up, mechanically mixing it with the acid and with the pepsin, and to digest the food. And then the pyloric sphincter occasionally opens for a brief minute or two, well, probably less, maybe just a few seconds or a minute, so that some of the chyme that has reached the pylorus gets squeezed into the duodenum at small bursts of so a small bolus of chyme gets released into the intestine. So let's look at this. So this is an endoscopy of a camera that was inserted in somebody's stomach. Clearly the stomach was emptied of all contents because there's no chyme in there. It's the shiny stuff that we'll see is the mucus lining the stomach. And what we're seeing, so they must have stimulated the stomach to undergo the peristaltic actions even in the absence of food. Somehow they stimulated that, and I'm not sure I know exactly how they did that. Probably by probably a cholinergic agonist or some other hormonal agonist to stimulate gastric motility and secretions even in the absence of food. Can you see it? So this is now a wave of peristalsis, sort of, we're looking down the stomach, so there's a sort of a closure of a thing that is now squeezing chyme towards the pyloric sphincter and then opening up to sort of allow it to mix, you know, so some of it is going forward down the pylorus, which you can just barely see there down at the bottom, and some of it is then getting sort of squeezed back up and for another cycle of squeezing. So sometimes I show this before Thanksgiving, but students don't usually appreciate that. So there's a number of these, but let's go back. So what you're seeing is a wave of peristalsis in your stomach. Who knew that you had something so alien looking inside your body? There's this wave of peristalsis squeezing a bunch of bolus of food, chyme, now pushing it down towards the pylorus, and then opening it up so that it's mixing with the content that's towards us. Some of it is going to get squeezed out through that pyloric opening, through the pyloric sphincter. And so these waves of churning action mix the chyme and squeeze it out through the pyloric sphincter in small bursts. Okay, so this is an image from your textbook that shows, you know, this basically shows what we saw in the video, squeezing a bunch of stuff towards the pylorus and then opening. Some of it goes out the pyloric sphincter and some of it gets churned back into the stomach. Okay, so now that we're in the stomach, the food is getting mechanically mixed with acid. Now we're, yes, Zoe.
Just really quickly, so the sphincter pushes stuff into the duodenum, but there's another sphincter from the esophagus to the
Right.
stomach. Does that also do the same thing of like?
So the sphincter, the lower esophageal sphincter, only opens in response to a bolus of food moving down the esophagus, at least normally. So during the churning action in the stomach, the lower esophageal sphincter stays closed so that the chyme does not regurgitate back into the esophagus. We do not want stomach acid to flow back up into the esophagus. That's usually harmful to the esophagus, that causes heartburn. And so the lower esophageal sphincter stays closed unless a bolus of food is moving from the esophagus into the stomach. So the lower esophageal sphincter is either sealed by the sphincter or it's sealed by the bolus of food moving through the sphincter so that acid does not regurgitate. So let me have the laser, please. So here's a cartoon of the stomach. What we're doing is now zooming in on an area of the stomach, looking at the folds, the rugae of the stomach, and the layering. So some of these layers you know from before break. We talked about the epithelial cells, the lamina propria. So I'm using some of these names that were on a slide from before break. So a layer of epithelial cells, lamina propria, muscularis mucosa, and then circular and longitudinal muscle, Mixed in here are the submucosal and myenteric nerve plexuses that control the contractions of the muscle. It's these muscles that do that peristalsis of the stomach. Now let's, okay, so I'm reminding you that there's a layered structure, the names of which are names. That's anatomy. Inside the stomach, these downward loops are called crids, or gastric glands. So let's use the word that's in the slide. Gastric glands in the body of the stomach. Corpus is just Latin for body. So if you call it the corpus, that's okay. The rest of us can call it the body. Anyway, these glands of the stomach have a variety of cells that release the material necessary for digestion. So let's go through those cells. So a generic epithelial cell. This tooth-like structure is supposed to be the apical membrane. Remember in the kidney, the apical membrane was folded. We called it the brush border in the kidney. Same structure here. This is the apical membrane of a epithelial cell, and this is the basolateral membrane of an epithelial cell. So basolateral, apical. Basolateral, apical. Basolateral, apical. Okay, so they're all epithelial cells with the standard structure. Remember, the apical membrane faces the lumen, and the basolateral membrane faces the lamina propria in this case. So, epithelial cells. We have cells that secrete mucus. Remember, mucus is spelled, I won't grade you wrong for spelling, but just to be clear, mucus is released by mucus cells. Okay? That's the correct language. Mucus is the material, mucus is the name of the cells. So mucus cells secrete mucus. And mucus here is to protect the lining of the stomach from the acid that we're going to talk about in just a moment. There are stem cells. The lining of the stomach is a rich niche for stem cells, because the cells of the stomach are constantly getting attacked and degraded by the acid in the stomach. So the lining of the stomach is constantly being regenerated by stem cells. So there's a rich population of stem cells in the stomach to recreate the cells as they're being destroyed probably by every single meal. The parietal cells are the cells that secrete the acid. So these are the cells specialized for acid secretion, and we will get to the mechanisms, the molecular mechanisms of acid secretion in a few minutes. Parietal cells. Chief cells are the cells that secrete pepsinogen, the precursor to pepsin, which is the enzyme that the stomach uses to start to digest protein. So mucus cells secrete mucus, parietal cells secrete acid, and chief cells secrete pepsinogen. There are also endocrine cells, and some books call them enteroendocrine cells, that secrete hormones. Well, a hormone, a specific hormone called gastrin. Gastrin is a hormone secreted by the endocrine cells of the stomach. So we're going to start to mix up some endocrinology into digestive because it's important. But unlike all the other things that we've been secreting, remember the mucus is being secreted into the lumen of the stomach. So it can protect the surface of the stomach, which it does somewhat. The acid, it being secreted by the parietal cells, is being secreted into the lumen for digestion of food. The pepsinogen is being secreted into the lumen of the stomach for the enzymatic breakdown of protein. Hormones are secreted by endocrine cells into the lamina propria because they are going into the bloodstream, not into the lumen of the stomach. Hormones, we don't want to put our hormones into the lumen of the stomach because they would get broken down. we want to put our hormones into the bloodstream. So they secrete, but their secretion is into the blood flow, blood supply. Remember, the lamina propria has blood vessels that get the hormone that is secreted by the endocrine cells in the gastric glands. Okay, so these are the cell types that secrete hormones into and other things, mucus, acid, and carcinogen that is necessary for digestion. Zoe?
Is that also like ghrelin and like the?
Ghrelin and leptin and ghrelin are other hormones that are secreted by the hypothalamus that control appetite.
Right.
So we are not at the high level of control yet.
Okay.
We're at a very basic level when we talk about gastric.
Okay, so it's not, that stuff isn't actually in the stomach, it's somewhere
No,
else.
that's right. So that's in the brain.
Yeah.
Appetite is a neural thing, not a stomach thing. Yes. You know, I don't even know. Not that I know of. Not that I know of. The superficial epithelial cell might be an absorptive cell for absorption of things like water. Because the stomach the stomach doesn't do a lot of absorption. Most of the absorption is going to be in the intestine, the small intestine. But there could, you know, when you drink water, some of it is absorbed by the stomach. So I'm not sure what the superficial epithelial cell's function is. But I am sure about the rest, and that's what you should focus on. Okay, so remember, this is just an electron micrograph. Remember that these cartoons showing these tiki things are cartooning something real, the highly folded brush border of the apical membrane. I guess in the kidney we call it the brush border. In the digestive tract we call it microvilli. So this is a cell, this is a lumen, this is an intestine, but the same thing is true in the stomach. Microvilli lining the apical membrane of a epithelial cell. So this is the beginning of the basolateral membrane of this cell, and separating this cell from this cell. Okay, so you remember mucus. These are the same cells that secrete. So in the respiratory system, we call them what? What kind of cells secrete mucus in the respiratory system? Goblet cells. Same type of cells, except that here in the digestive tract, we don't call them goblet cells, we call them mucus cells. I don't know why. I have no idea. Do you remember what mucus does, or what mucus is? Here's a video that shows so that is mucus. That's mucus being secreted by a hagfish, being mixed with water. Mucus being secreted, hydrated, turning into mucus. So mucus is a mixture of mucins that are secreted by the mucus cells and water, making that lovely material. And that's enough. So here is a globule of mucin being secreted by a mucus cell in the stomach, secreting a packed globule of these mucin proteins that then mix with the water, the water and acid of the lining of the stomach to make mucus that then coats the inside of the stomach, trying not completely successfully, but at least partially, to protect the apical surface from the acid hydrolysis of that membrane. I'm not gonna ask you to know the structure of mucins, but they're polysaccharides. Okay, let's get to acid secretion. So mucus secretion is simple exocytosis of globules, of packets of mucins. And you know all about exocytosis. You learned about it in cell phys, and we don't need to review it, but remember there are snare proteins and other things important for exocytosis. Now let's talk about the mechanism of acid secretion. And in fact, you've already seen this in the kidney. Which membrane is this? It doesn't give you very many clues, but it gives you enough to tell you which membrane is this one. That's the apical membrane. How did you know? Because it's facing the lumen of the stomach. This and this are the basolateral membrane facing the lamina propria that is vascularized with capillaries. So, okay, what type of cell are we in? We're in a parietal cell. I was going to ask you that, but it's right there. Parietal cells are the cells that are specialized for secretion of acid. The acid that we secrete comes from carbonic acid. So you've seen this before. Carbon dioxide, which is at 40 millimeters of mercury partial pressure normally. Carbon dioxide diffuses everywhere. Remember, it's in the blood. It's in arterial blood at 40 millimeters of mercury, but arterial blood is perfusing these cells. So, and since carbon dioxide diffuses everywhere, the partial pressure of carbon dioxide in parietal cells is, I don't know if it's 40 millimeters of mercury, but there's carbon dioxide there. Carbonic anhydrase catalyzes the conversion of carbon dioxide into carbonic acid, which dissociates. This is a story that you've seen before. You know this. The acid, the proton of carbonic acid, let's just forget, I don't even know what point your book was trying to make here. The point is the proton of carbonic acid is pumped out of the apical membrane by a proton ATPase. It happens to be a proton potassium ATPase that pumps protons into the lumen of the stomach and pumps potassium ions into the cell, sort of like the sodium potassium ATPase. But the business end of this ATPase is the primary active transport of protons. There is also there's passive transport of chloride through chloride channels. And this is just a passive flow following the pumping of a cation. The pumping of a cation is accompanied by a rich passive flow of chloride. And actually water to some extent too, because we're pumping a solute into the lumen of the stomach. So hydrochloric acid is pumped into the stomach. There's the hydrochloric acid being pumped into the stomach. The potassium is sort of going through one of those feudal cycles that just happens to be there, but that's not the functional thing to focus on. Protons, primary active transport, chloride, passive transport, hydrochloric acid is pumped into the stomach. The bicarbonate is gotten rid of by the bicarbonate chloride exchanger on the basolateral membrane. You've seen all the pieces of this before. It's just a matter of putting them together into the parietal cell story. Any questions about this?
Wait, so that chloride is a channel or is it?
This is a chloride channel, and this is a bicarbonate chloride exchanger, secondary active transport. Okay, let's do a clicker and then maybe we'll wait for it. stomach secretes acid during the gastric phase, and so we'll talk about the phases of digestion in a minute. The stomach secretes acid during the gastric phase of digestion. What happens to plasma pH? Blood pH. Okay, I see a lot of correct answers. Blood pH increases, right? Because we are secreting acid into the stomach, and we are pumping bicarbonate into the blood. Pumping of bicarbonate would make the blood more basic, that is to say, a higher pH. Okay, so let's do a Davenport diagram here. Okay, so anybody, I'll pass the laser if you force me to, but I'll take volunteers instead. What is the ordinate? Bicarbonate. Concentration of bicarbonate in the blood. Units of millimolar or milliequivalents per mil, per liter. 26 millimolar is a normal value. What's the x-axis? pH. Blood pH. Plasma pH. 7.4 is a normal value. So here we are at the normal value. I think most of you did pretty well on this, I would say. We are breathing normally. So we are on this curve, how do we label this curve? Partial pressure of CO2 in arterial blood is 40, what are the units? Millimeters of mercury. Okay, so here's where we are before we start our meeting. Okay? This is the normal starting condition. What arrow do I draw to indicate what happens during the gastric phase of digestion? Can somebody describe an arrow? Or come up here and just draw it. It is alkalosis. Okay, so we're going to move in this direction because that's alkalosis. I'll give you two choices. Am I going to move like this? I'll give you three choices. Am I going to move like this or like that? Okay, let's do this as a clicker. A, B, C. Because it's alkalosis, we already know that the pH increases, but now which of these is the correct answer to describe the alkalosis that happens during the gastric phase of digestion? Before any compensation. Right, so most of you are getting it all right. We are breathing normally, so we're still on the normal curve. So this is metabolic alkalosis during the gastric phase of digestion. Okay, so now let's talk about the renal. What would the renal response be if we had no respiratory compensation? What would the renal response be? Somebody describe the arrow. No respiratory compensation. I'm separating out respiratory from renal compensation for this metabolic alkalosis. Which way is the arrow?
We try to reestablish the blood pH.
It would try to reestablish blood pH. Is it going to do it? Okay, I'll give you three choices. A, so we move to this point, A or B. A or B for the renal response to the gastric acid metabolic alkalosis. I mean the gastric phase metabolic acid alkalosis. Right, you're getting it right because at B is the correct answer. We are still breathing normally, so we're still on this curve. So the kidney is going to try to excrete some of that bicarbonate that the stomach has produced. And you know mechanisms for increasing the excretion of bicarbonate. Down-regulating carbonic anhydrase, down-regulating the proton pump in the kidney, in the renal tubule. Both of those would tend to increase the excretion of bicarbonate. And what is the respiratory? Now let's say we had respiratory compensation, but no renal compensation. Which arrow? Respiratory compensation, but no renal compensation, would show up on this curve as what? Somebody describe that arrow. You know, so respiratory compensation. We have gone to this point, but we're not doing this, at least in this condition now. We've gone to this point. What is the respiratory compensation? Anyone? Down this way? Down this way? Is that going to help us restore blood pH if we go down this way? It's going to go up this way to a new curve, because now are we hyper or hypoventilating? Well, that's bad language. Are we increasing or decreasing ventilation to try to normalize this pH in this condition? Decreasing. We are going to a PaCO2 that is less than 40 millimeters of mercury. I'm sorry, greater than. We are hypoventilating. We are increasing the carbon dioxide in order to rebalance the pH. Remember, the pH was a function, there was a complicated function of the ratio of CO2, or bicarbonate, and CO2. Remember, there was the Henderson-Hasselbalch equation had a term that was this ratio. If we increase bicarbonate during the gastric phase of digestion, the respiratory response is to increase the carbon dioxide concentration so that the pH becomes re-normalized by decreasing ventilation. So after a big meal, during the gastric phase of a big meal, there is a period of hypoventilation. Now, if you're aware of it, it's probably because your stomach is so full that you can't actually, that's not the hypoventilation that we're talking about. But it in fact is adding to it, right? If your stomach is so full that your lungs are not ventilating thoroughly, that would lead to hypoventilation. Okay, so once more, the Davenport diagram now, adding in the digestive system during the gastric phase. Okay, let's take a short break, and we'll come back to continue digestion in the stomach. Pepsin and pepsinogen. So remember that parietal cells are the cells that secrete gastric acid. Chief cells are the cells that secrete pepsinogen. Pepsinogen is a precursor to pepsin. And the acid hydrolyzes the pepsinogen into pepsin, which is the active protease. So pepsin starts proteolyzing protein. It also proteolyzes pepsinogen. So there's a positive feedback loop going on here. Pepsin hydrolyzes protein into small oligopeptides, and it hydrolyzes pepsinogen to make more pepsin. So the acid gets the story started. The acid starts this positive feedback loop, and then this positive feedback loop goes all the way until all of the secreted pepsinogen is converted into pepsin, and so we have a maximal amount of pepsin that hydrolyzes protein into smaller oligopeptides. This is not the end of the story for protein digestion. We're going to see 3 more enzymes when we talk about the intestines on Thursday. But this is the start of the protein digestion story. Remember, we didn't have any enzymes to digest protein in saliva, but now we have an enzyme, pepsin, to start the enzymatic hydrolysis of protein in the stomach. So now let's talk about control of these processes. And specifically, right now, we're talking about the control of acid secretion. So your stomach does not secrete acid constitutively. It's not in your best interest to be pumping a bunch of acid into an empty stomach. That would degrade the mucin, that would degrade the protective layer of the mucus, and that would cause acid hydrolysis of the membrane, which would lead to ulcers, which we're maybe going to get to in a few more minutes. So acid secretion by the stomach is under tight control. And it's controlled by stimulatory hormones, or neurotransmitters, and several inhibitory hormones and hormones. So the idea here, so this is a very poor cartoon from your textbook of a parietal cell. It should be boxier, right, because it's an epithelial cell. It should have a clear apical membrane and a clear basolateral membrane, right? It's a parietal cell. It should look more like, oops, going the wrong way. It should look more like that, or ideally, or that. That's a pretty poor one. Or like that, a parietal cell. There's a pretty good cartoon of a parietal cell with an apical membrane and a whole bunch of vesicles inside the cell ready to be exocytosed into the apical membrane, delivering the proton pump to the membrane. So now let's go to the wrong way. No. Okay, so now we're going to the apical membrane. This is the apical membrane of the parietal cell, and this is the basolateral membrane. Apical membrane is where the proton pump pumps acid into the lumen of the stomach. There's the proton pump. So here's the apical. This slide makes me crazy because I have to spend a lot of time getting everybody oriented. But here's the apical membrane, and there's the basolateral membrane. The basolateral membrane, in this case, has receptors for neurotransmitters and hormones that cause the exocyte. So the proton ATPase is in intracellular vesicles. So between meals, when we do not want to be pumping acid into the lumen of the stomach, the vesicles contain the proton ATPase, waiting for a trigger to get exocytosed into the apical membrane, delivering all this pump, which will then start pumping acid into the lumen of the stomach. So the pumps are pumping acid into the lumen of the vesicles constitutively, where it can do no harm. And then during a meal, we stimulate the exocytosis of these vesicles into the apical membrane, where that pump can now start pumping acid into the lumen of the stomach rather than into the lumen of the vesicles. Okay, so this is a slide from your textbook that describes how three hormones, let's talk about the three hormones that stimulate the exocytosis of these vesicles into the apical membrane, delivering the proton pump that we want for pumping protons, acid, into the lumen of the stomach. So your textbook has this extremely unhelpful arrow that describes a very complicated process between a receptor or a set of receptors and exocytosis. So it will come as no surprise to you that let me just, now this is where we're going. A more complicated roadmap between receptors and exocytosis of vesicles into the apical membrane. But before we get there, let's just talk about some of the triggers. So, and now we're going to talk about 2 of the 3 phases of digestion. So just talking about food right now, just talking about food, we are initiating a cephalic phase, cephalic head, a brain phase of digestion. You're thinking about food. You might even be salivating a little bit more than you were a few minutes ago before we started talking about food. And I can do it, you know, like I did before break by talking about gravy and turkey and lasagna and whatever. The brain is triggering neural activity that releases acetylcholine. So acetylcholine, this arrow right here, is acetylcholine from the nervous system stimulating the parietal cell. So this arrow represents the arrow from here, from an acetylcholine receptor to this exocytotic event. Neural activity in the cephalic phase, thinking about food. Acetylcholine is the neurotransmitter. Receptors on the parietal cell are going to stimulate the exocytosis of vesicles containing those pumps to pump acid into the stomach. So there's the cephalic phase flow chart. In addition, there's stimulation of the nervous system stimulates the endocrine cells of the stomach to secrete gastrin. So acetylcholine stimulates gastrin secretion from the endocrine cells of the stomach. Remember, we had the endocrine cells in the gland. They have receptors to acetylcholine, and when they bind acetylcholine, they release gastrin into the bloodstream, and the bloodstream then delivers gastrin to the parietal cells of the stomach. And so now this arrow is gastrin stimulating exocytosis. Acetylcholine, gastrin. Acetylcholine stimulates histamine secretion from mast cells that are in the gastric mucosa as well. So histamine is another positive stimulator of the exocytosis of these vesicles, the delivery of the proton pump, and the pumping of acid into the lumen of the stomach. So we have to talk about acetylcholine direct and indirect effects, histamine's direct effect, and gastrin's direct effect on the parietal cell itself. Yeah?
Where is the study you're using now?
It's from other endocrine cells in the stomach. Sort of on the outside lining of the stomach. So there's a whole set of endocrine cells, G cells that secrete gastrin, S cells that secrete, or H cells that secrete histamine, S cells that secrete somatostatin. I'm not going to ask you to know the names of those cells. These are endocrine cells that secrete these hormones. So that's gastric phase. That just happens when we think about food. When food actually enters the stomach, now we're talking about the gastric phase, which continues this process that was triggered by the thinking and, frankly, the swallowing of food. So now we have food in the stomach. It's becoming chyme. Food in the stomach food in the, here we are, food in the stomach, gastric phase, stretching of the stomach, amino acids in the stomach, peptides in the stomach, a decrease in the acid in the stomach, because after all, if we pump food into the stomach, that acid is now being used, being mixed with food, so the pH of the stomach goes up, just because we're diluting the acid that was there before. And so all of these are gastric phases, stretching of the stomach, peptides and amino acids in the stomach, frankly, some carbohydrate in the stomach, anything that stretches the stomach triggers gastrin secretion, and now we have more gastric acid secretion by the parietal cells. So most of this slide is looking at the cephalic phase, but here is the gastric phase. Material in the stomach, food in the stomach, triggers gastrin release, and gastrin is going to stimulate parietal cell exocytosis, increased pumping of acid into the lumen of the stomach. Acid in the stomach, low pH, actually stimulates, this is a negative feedback loop here. Somatostatin, if the pH in the stomach gets too low, like between meals, somatostatin inhibits all of this process in order to prevent more acid from being secreted and allowing the endocytosis of those vesicles to reverse this process here. So somatostatin doesn't just inhibit this arrow, it stimulates, and you should illustrate this in your slide. Did I give you this slide as a handout?
Yes.
Somatostatin stimulates a reverse arrow, a reverse process of endocytosing this membrane back into vesicles. Remember, endocytosis and exocytosis are, work against each other. They're both reversible. So, understanding the flow chart of the cephalic phase and the gastric phase is important. And here is a slide that it looks impossible, but you know everything. You know how to interpret this. Let's walk through this. Here's gastrin. Here's gastrin. Gastrin is a hormone that stimulates exocytosis of these vesicles to make more pump in the apical membrane. So gastrin activates a gastrin receptor coupled to GQ. You should be off and running now with the GQ story. GQ, phospholipase C, hydrolyzes phosphoinositol bisphosphate, PRP2, into IP3 and diacylglycerol, calcium release, activation of protein kinase C. Activation of protein kinase C increases the exocytosis of vesicles and increases the expression of the proton pump in the apical membrane. So here is the gastrin story right here. Here is the acetylcholine story. It's the same story, it's just a different receptor. The acetylcholine interacts with another, a different GQ coupled receptor. Here's histamine. Histamine also stimulates, but not through PKC, but through GS and PKA, the canonical GS story. Right? I'm not even walking through this one because this one is so second nature to you by now. You know it by heart, forwards and backwards. So here are the stimulatory receptors: gastrin receptor, acetylcholine receptor, histamine receptor, that through GQ or GS, through PKC or PKA, increases gastric acid secretion. Somatostatin works through a GI to inhibit the GS story, inhibits adenylyl cyclase, and allows a constitutive endocytosis of these vesicles, removing the pump from the apical membrane. We're not going to talk about prostaglandins. So what looks like a completely horrible slide is really a slide that just builds on your previous knowledge. I'm not going to ask you to know the name of the gastrin receptor. I'm not going to ask you to know the name of the acetylcholine receptor, but I'm happy to use that language if you want. So your textbook has a series of tables which I think are useful, and I recommend that you study them. We're not going to go through them here in class very much, except that I want to, it talks about the cephalic phase, sight, smell, taste, and chewing of food. Here is the enteric nervous system. Parasympathetic nerves means it's acetylcholine, releasing an increased acid secretion. We've talked about that already quite a lot. Here is the gastric phase, stretching of the stomach, peptides in the stomach, decrease in proton concentration because of dilution of the gastric acid, increasing acid secretion. And then we'll get to the intestinal phase Thursday. So your book does a good job with these tables, and I want to point out this one, which I gave you as a handout, but I know you can't read because it's in your textbook. So what we're going to see on Thursday is that, so here's the gastrin story. It's a peptide hormone secreted by the stomach, stimulated either by the gastric phase or by the, by the cephalic phase or by the gastric phase. And it stimulates acid secretion and motility of the stomach. It stimulates digestion in the stomach. Let's not get into these things just yet. On Thursday, we're going to talk about two more hormones that are essential for pancreatic secretions and also essential for inhibiting stomach secretions. So I'm just introducing two more words that we're going to be talking about on Thursday. CCK, which stands for cholecystokinin. CCK. Cholecystokinin is a hormone. We'll talk about it. It's released from the intestine. It's important for secretion of things from the pancreas. So we'll talk about this on Thursday. But what I want to point out is that these hormones, in addition to somatostatin, inhibit the secretion of acid from the stomach. So once the food is getting into the intestine, we now have an inhibitory stimulus that says, okay, food is getting into the intestine, let's shut down, we've got an intestinal phase coming up, let's start to shut down the gastric phase. And so we're starting to turn off the stimuli that the gastric phases and the cephalic phases turned on in order to not overdo the acid production in the lumen of the stomach. Okay? So I recommend studying these tables and understanding them for the quiz, especially the ones that I just walked through. Don't have to do everything. Let's not get to liver until we talk about it. Okay? We'll start on Thursday with the clicker and then get into the gastric ulcer story, which is an amazing scientific story, and then get into the intestine and the Thursday.